So I'm going to use this diagram to illustrate the two forces that govern the movement of water out and into the capillary. Okay. So let's say this is the capillary, and dash lines means indicate variable length. Okay, so this is at the start of the capillary, which is attached to which is continuous to the arterial. So this is called arterial end of capillary. And this end of the capillary will join back to the venule. So this is a venous end of capillary. So in the arterial end of capillary, there are two forces that act on the water, which is the blood plasma inside it, which eventually cause the blood plasma to leak out from the capillary through the gaps between the cells at the wall into the tissue fluid. So the two forces are represented by these two arrows here, which for me, I actually use the symbol so that you can remember better, which is psi, okay, equals to psi S plus psi P. Actually, I want to write psi P first. In this case, it's better to write psi P first, okay? Psi P plus psi S. Okay, so this arrow here represents the effect of psi P on the movement of water molecules. Okay, this arrow here represents the effect of psi P on the movement of water molecule, and this arrow here represents the effect of psi S on the movement of water molecules. Okay, so let's look at the psi P first. Psi P inside the blood vessel is the hydrostatic pressure. Okay, this psi P is due to the hydrostatic pressure inside the blood due to the pumping of the heart, which I mentioned already yesterday. Now, of course, other than pumping of heart also, there are other pressure that may also be generated due to elastic recoil, okay, of the elastic fiber, as well as the contraction of the smooth muscle. So smooth muscle contraction. So all these three can result in hydrostatic pressure, okay, that can increase hydrostatic pressure of the blood. But the main one, of course, is the pumping of the heart. So because it's at the arterial end, the pressure is very high. So it tends to push the blood to leak out from the pores. It's just like you got a plastic bag filled with water and you start to poke the wall of the plastic bag. And then when you squeeze the plastic bag, means you're actually creating hydrostatic pressure. When you squeeze the plastic bag, you're creating the hydrostatic pressure, and therefore the water will leak out from those tiny little pores that they've created just now, just like this one here. Okay, so hydrostatic pressure in the blood can cause the blood plasma to leak out. Okay, but don't forget, inside the blood also there are a lot of solutes. So therefore, the solute will reduce free water molecules, correct? Because they attract water molecules around it, and therefore there'll be less free water molecules. So that will be increased solutes, reduce solute potential, so reduce water potential. So the solute that are found inside the blood vessel will tend to reduce the water potential. So therefore, it will drive water to actually move into the bloodstream. It will drive water from tissue fluid to move into the blood plasma because of a lot of solutes here that tend to decrease water potential. Okay, so there is a force that tend to draw the water from the tissue fluid into the blood. So there are two forces acting on the blood plasma. So therefore, when you add these two forces together, overall hydrostatic pressure that pushes the water out is stronger than the the solute that tend to draw the water backwards. So therefore, there's a net movement of water out from the blood vessel. Okay. Now, this is in the arterial end. In the venous end, now what happened in the venous end is two things. Now, the blood has flowed through the capillary now, so the blood pressure will drop as the distance that it travels becomes further and further away. So the longer the distance is traveled, the more resistance it has encountered. So therefore, the pressure will drop. Okay. So as it increases the distance that it travels, there'll be more resistance that it has encountered. So therefore, the pressure will drop. So therefore, the pressure has dropped until significantly. Can you see that it's lower already now? So much lower than original one. So the hydrostatic pressure that can push the blood plasma to flow out becomes so much reduced already. But what happens here yesterday, remember I mentioned that when ultrafiltration occurs, it's like you're pouring the jar of Ribena, okay, to the glass through a sieve. Let's say, for example, you put a sieve in the glass here, okay? So you pour the Ribena through the sieve. So whatever you have here, your Ribena concentration, after ultrafiltration, your Ribena concentration in your glass and in the jar is actually the same because it originates from the same place. So they are the same. With one exception, the larger protein, where these proteins are soluble, but because it is larger than the pore size of the sieve, so this sieve here got pore. So those large proteins are larger than pore size of the sieve. They will not be able to pass through during ultrafiltration. So they remain inside the jar. Okay, say for example, they remain inside the jar. So as you pour more and more liquid out of the jar, your solution in the jar will become more and more concentrated, correct or not? Because you got more water flow out, but your number of these protein molecules, the large protein, remain the same. And of course, the rest of the solutes are still there. So it becomes more and more concentration. So when it becomes more and more concentrated, that means when it's even more concentrated, the solute potential becomes even lower. So the water potential becomes even lower. So therefore, its ability to drive, so because it is even lower, the concentration gradient between tissue fluid and the blood plasma becomes even steeper. So there'll be more water that will be pulled to go back into the blood vessel down the water potential gradient. So overall, when you add these two forces together, effect of hydrostatic pressure that push the blood plasma out, and effect of the solute that tend to draw the water in, overall, there'll be net movement of water back into the capillary from tissue fluid. So that's how the tissue fluid can flow back into the blood plasma. So at the start of capillary, it will flow out because it's supposed to carry oxygen, nutrients, hormones to the tissue. And then at the end of the capillary is when 100%, so when, let's say for example, so this is the volume of the blood plasma that leak out, okay? The volume of blood plasma that leak out. So out of 100% that leak out, approximately 90% will go back into the blood plasma at the venous end. Okay, 90% will flow back, driven by this water potential gradient. Actually, both of them are driven by water potential gradient one. Okay, but you need to describe, mainly this one is driven by hydrostatic pressure, and this one mainly is driven by reduce in hydrostatic pressure and increase in the concentration of the blood plasma due to large protein that cannot come up. So that causes water to flow back into the blood plasma due to the large protein. Okay, the large protein that cannot flow out, therefore increasing the concentration of blood plasma here. Okay. So have you seen edema? That means that the part of your body that is becomes swollen. You know, sometimes you got infection and then the part of infection becomes swollen. So that's called edema. Okay, edema. Edema is a swelling of tissue due to accumulation of excess fluid. Okay, so usually, okay, I forgot to say, you see it 100% flows. Okay, this is capillary, yeah. So if 100% comes out here and only 90% goes back in, you're left with 10% of the water, right? That still remain in the tissue. So this 10% must be drained out through another system, which is called the lymphatic system. So this 10% will drain through the lymphatic system, okay, which is made out of blind-ended lymph vessel. So therefore, 100% come out. Okay. 90% goes back in to capillary, 10% drain into the lymphatic lymph vessel. Okay, so this is capillary. So usually if this process happen efficiently, we will not expect to see that any part of tissue becomes swollen for a long time. Okay, but if the tissue becomes swollen for a long time, that means something must be wrong already in our body. The excess fluid starts to accumulate in our tissue. It doesn't go back to capillary or it doesn't flow into the lymph vessel, and therefore it accumulates in our tissue becomes swollen. So it's called edema. Okay, so why is it this can happen is there are two possible reasons. First reason is the protein concentration in the blood is very low because this person is malnutrition. Okay, malnutrition, cannot get enough nutrients, especially protein. So when you don't have protein in the venous end, then you cannot increase its concentration, you cannot reduce the water potential. So you cannot produce a water potential gradient to drive the water to go back. So that's why the water remains inside the tissue, because no protein or very few protein to actually increase the concentration of the blood plasma in order to reduce water potential. So therefore, the gradient is not steep enough to drive the water to go back. Okay, so when you got low blood protein, psi-S will increase, psi will increase. So the water potential gradient is not steep, so water cannot flow back to the capillary. So therefore, water collects in the tissue and then the tissue will swell, so it's called edema. Okay, or second reason is because you have got high concentration of protein in the tissue. So, you know, tissues are made of cells, right? Okay, so tissues have got a lot of cells inside here. So those cells here might be secreting protein. We have got cells that they are supposed to secrete protein one. Okay, for example, they secrete glucagon or insulin, antibodies, and so on. So when they secrete this protein, and this protein remain inside the tissue fluid and is not being drained away. Okay, why they're not drained away? Probably the lymph vessel is blocked by something inside here. So the liquid here doesn't flow. So when it doesn't flow, of course, the protein will not flow. Because all solute flow because the water moves and carry the solute with it. So when something blocks here, the water does not move. So when the water does not move, the water in the tissue fluid cannot go in and therefore cannot carry those protein into here. So protein remains here. So when the protein remains here, it will increase the solute concentration. So therefore, it will also reduce the water potential. So therefore, it will create a concentration gradient for the water to move in. So water continues to move in until equilibrium is reached. So this region here will fill with water. So it becomes swollen. So this is called edema. Okay. So this is a very nice diagram that I saw yesterday. Very, very nice. So this is a capillary. Can you see the nucleus of the endothelial cell? The squamous endothelial cell has got very flat, elongated nucleus, right? Flat, elongated nucleus. And then this pink color region here is the cytoplasm of the cell. Can you see not? This part only. This is the cytoplasm of the cell, right? This is cytoplasm of the cell. So can you see that capillary will bring oxygen and nutrients very close to the surrounding cells of the tissue so that the rate of diffusion will be very fast. The rate of exchange will be very fast between the blood and the cells of the tissue. So this is what I want to illustrate using this diagram. Okay. So this diagram here illustrates from the bloodstream, the blood plasma will leak out from the bloodstream due to high hydrostatic pressure, and the exchange of materials will occur between the tissue fluid and the cell. And after that, the excess fluid, etc., will flow back to the bloodstream as well as to flow into the lymph vessel. Larger proteins that cannot go into the bloodstream because it's larger than the gap, it will go into the lymph vessel because the gap here is bigger, and then be transported and then eventually join back to the bloodstream at the subclavian vein. Okay. Now, after this, the rest of the
Teacher, you are muted.
Teacher, we cannot hear you, teacher.